DNA has got two big jobs, and we have talked about the first one, which is DNA replication. This is a separate event, right? When it comes to DNA replication, a cell will start with one copy of all the DNA that you have. It will make a second copy of that DNA, and then the cell will divide so that two new cells, each with their own copy of DNA, will be the result. That's what will happen. That's when DNA replication happens. Now, I've got one more animation for you to look at about DNA replication. The other one was so pretty. I really like it. But let's look at this one, right? So we are looking at some DNA. Let me pause this for a moment, okay? We are looking at some DNA. You'll notice that they've colorized one strand blue and they've colorized one strand red. What is this coming in here? That is going to be one of our enzymes that you need to know. Here we've got an enzyme, and this is the enzyme helicase. Helicase is going to unwind the DNA, and then it'll be topoisomerase that holds the unwound strands in their open position, right? So here's helicase. Helicase is an enzyme. You know it's an enzyme because it ends with the letters ASE. So helicase is breaking the hydrogen bonds between the base pairs. Remember, if over here is an A, that would be a T. If over here is an A, that would be a T, right? Now, what are these other proteins? You don't actually need to know their names, but there are special proteins that keep everything open so it doesn't close up again, kind of like keeping the Velcro open. Now, let me pause for a second. So helicase unwinds and opens the strand. Topoisomerase keeps them from over twisting. What is this? This is DNA polymerase. Remember, anything that ends in ASE, that's an enzyme, and the rest of the name should tell you what it does. What does this enzyme do? It makes a DNA polymer, right? Remember, DNA is a good example of a polymer. It is made out of monomers called nucleotides. Here's a nucleotide. Remember, phosphate sugar base. This base is guanine. Here's a nucleotide, phosphate sugar base. This base is a thymine. And so the DNA polymerase is going to walk down this strand of DNA, and everywhere it finds a C, it'll say, oh, I guess I better put a G on the opposite side. DNA polymerase is about to get to where there's an A. So it will say, I need to put a T on the other side, right? So here it's going to go ahead and put the opposite or the complementary nucleotide. So there are actually two different strands. We're just thinking about the first strand. The second strand it's a lot harder to make the other side of DNA. With the other side of DNA, you need RNA primer and you need primase, and it's a lot harder. It's an interesting story, but it's not necessary for you to learn the other story for AP 120. When you get to microbiology, if you're on a pre-nursing track, you're going to need to know that. So one strand is easy to copy and one strand is hard. We're just looking at the DNA polymerase that's working the easy way. The helicase has opened it up. Topoisomerase is keeping it open. The DNA polymerase is just going, oh, that's a G, I'll put a C. Oh, that's a C, I'll put a G. Just chugging its way on down the line. It's got a very easy job to do. So you need to know helicase, topoisomerase, and DNA polymerase. This other part, it is the reason that first video I showed you on the other lecture had such a complicated image because of Okazaki fragments. Luckily, you don't need to know that for this particular class. Great. Now, that is one of the things that DNA needs to do. It needs to do DNA replication, but it's not the only thing. What is the second thing that DNA needs to do? It needs to participate in what is called transcription. Participate in transcription. All right? What is transcription? Well, with DNA replication, remember we started out with DNA, and when we were done, we had twice as much DNA. With transcription, we start off with the DNA, and when we're done, we will still have our DNA, but we will also have a little piece of RNA, right? Now, to make DNA, to do replication, we used DNA polymerase. In order to start with DNA and make a little piece of RNA, we need RNA polymerase, right? Remember, an enzyme, its names end in ASE, and the rest of the word tells you what it does. And RNA polymerase makes a polymer of RNA. It makes a piece of RNA. Now, we're going to be focusing on messenger RNA. All of the DNA that you have, it's enormous. And there are little tiny chunks of it that have the instructions for how to build a protein. Little tiny chunks. Each little chunk that says how to build a protein is called a gene. And when your nucleus wants to tell the ribosomes out there, hey, I want you to make this protein, here's how the nucleus does it. The nucleus will make just a little tiny copy of messenger RNA and sends the messenger RNA out of the nucleus. It's kind of like, when you think about it, all of your DNA is like an entire library of books. Okay? So let's imagine the Cerritos library is all of my DNA. And then if I wanted to do DNA replication, I'd have to build a whole new library. We're not going to do that. But it turns out that the library is going to tell me what I should cook for lunch every day. So I would go to the outside of the nucleus, the outside of the library, and whoop, here would come a little piece of paper out of the library, and it would be the copy of just one recipe from some cookbook in the library. And I'm supposed to make that recipe, and that's what I'm supposed to have for lunch. In this analogy, the library is all of your DNA. Well, the library is the nucleus. All the books are your DNA. What comes out in that little piece of paper? That's messenger RNA. And who am I? I'm going to be the ribosome. So the nucleus is going to tell me what to make. Transcription is starting with a small bit of DNA, a gene, and making an RNA transcript complement of just that gene. And it's only the RNA transcript that's allowed to leave the cell. So why do cells need this thing called transcription? Because the DNA stays in the nucleus where the DNA is safe. If that DNA gets damaged, we die. So we don't want the DNA to get damaged. The DNA never leaves the nucleus. Remember, the nucleus is like the executive office building in the factory. The genes are segments of your DNA, and they have the instructions to make a protein, right? Every gene, instructions to make actually a few proteins. So all of that's in the nucleus. Now the ribosomes, they're out there in the cytoplasm, right? Ribosomes could be attached to your rough endoplasmic reticulum, or they could be just floating around in the cytoplasm. But those are the things that are going to make the protein. So the instructions are in the nucleus, but the machines that are going to make the protein are outside of the nucleus. So we need to have the nucleus share the information with the ribosome. That little guy there, he's the ribosome, okay? And he's happy to build any protein that you want. You just have to tell him what it is. So the nucleus is going to just make a copy of just one little part of that DNA in the form of messenger RNA, and that is going to be the message. Messenger RNA carries the instructions of how to build a protein to the ribosome. All righty? Just as a review with the same image, replication happens when the cell makes a second copy of all of the DNA, and then the cell is going to divide, right? But DNA replication, sorry, we already did the DNA replication. DNA replication, you end up with twice as much DNA, and then you make a new cell, right? Transcription is going to happen when messenger RNA is made. Then what happens? There are two steps that are kind of joined together. Transcription makes the messenger RNA, and then the ribosome will take the instructions and read those instructions and make a protein. Making a protein is called translation, right? Translation, you kind of translate the genetic code of nucleotides, the GATC of DNA into the GAUC of RNA. You got to translate that into the language of amino acids so that you can make a protein. And by the way, this leads us to the three different kinds of RNA that we have. Ribosomal RNA, see that little R? That stands for ribosomal RNA. The ribosome is made out of ribosomal RNA and proteins. Write that down, okay? Ribosomes are made out of ribosomal RNA, rRNA, plus proteins. And it is going to read the messenger RNA, okay? The messenger RNA, it's the message. It is the actual little recipe. It's what was made by transcription. And then there's this other guy, transfer RNA. Technically, they help build the protein, and I will show you more about that in the next lecture.